Cell respiration! Woo! Glycolysis is the first step in cell respiration. It is a process for harnessing energy in which one glucose molecule is broken into two pyruvate molecules in the cytoplasm of a cell. All the reactions in glycolysis are also catalyzed by a specific enzyme. The first ATP attaches a phosphate group to one end of the glucose molecule. This creates glucose 6-phosphate. Glucose 6-phosphate then gets rearranged into fructose 6-phosphate.
The second ATP attaches to a phosphate group on the other end of the fructose 6-phosphate molecule. This phosphorylates fructose 6-phosphate into fructose 1,6-biphosphate. The backbone then splits apart to form G3P and DHAP.
DHAP is then converted into G3P, which results in two G3P molecules. This marks the start of the energy-releasing steps in glycolysis. The two G3P molecules give up one hydrogen and 2 electrons each, which reduces NAD+ to NADH. Then G3P combines with inorganic phosphate to form an unstable molecule called 1,3-BPG.
1,3-BPG then gives up the phosphate group to ADP, which forms 2 ATP through substrate-level phosphorylation. This results in a new molecule of 3PG. With this formation of ATP, the energy investment is paid off. The molecule of 3PG then rearranges to its isomer of 2PG.
2PG then gives up one hydrogen ion and one hydroxide ion, which forms water. As a result of this, a PEP molecule is formed. PEP is unstable and breaks down and releases enough energy to move one phosphate group to ADP, which forms another molecule of ATP. At this point, two pyruvate molecules have been formed. In summary, glycolysis creates two NADH molecules, two ATP molecules, and two pyruvate molecules for each glucose. After glycolysis, the two pyruvate molecules move from the cytoplasm into the matrix of the mitochondria with the help of a membrane carrier. This is where pyruvate oxidation and Krebs cycle occur.
Coenzyme A attaches to the acetyl group on the pyruvate molecule, which makes it not available to oxidize. At the same time, NAD+ is reduced and becomes NADH. The leftover carboxyl group is taken away as waste in the form of CO2. The resulting product is acetyl-CoA. The acetyl-CoA enters the Krebs cycle to continue aerobic respiration. Overall, this produces 2 molecules of NADH.
The Krebs cycle is a series of 8 reactions that transfers energy from molecules into usable forms for the cell. It also releases CO2 as a waste product. It occurs in the matrix of the mitochondria and is catalyzed by a specific enzyme for each reaction. The acetyl-CoA reacts with oxaloacetate, which already exists in the matrix, to form citrate. The CoA is then removed. This reaction is catalyzed by citrate synthase. The citrate then rearranges into its isomer isocitrate. This reaction is catalyzed by the enzyme isonatase.
Citrate is converted into alpha-ketoglutarate through a redox reaction. It does this by losing a CO2 atom and 2 hydrogen atoms that reduce NAD+ to NADH. This reaction is catalyzed by the enzyme isocitrate dehydrogenase. Alpha-ketoglutarate is converted to succinyl-CoA by binding with CoA. In the process, a CO2 is removed as waste and 2 hydrogen atoms reduce NAD+ to NADH. This reaction is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase.
Succinyl-CoA loses its CoA to become succinate, which produces major energy. ATP is then formed by substrate-level phosphorylation, which involves GDP and GTP, and CoA is released. This reaction is catalyzed by the enzyme succinyl-CoA synthetase. 2 hydrogen atoms reduce FAD to FADH2, and succinate is then converted to fumarate. This reaction is catalyzed by the enzyme succinate dehydrogenase.
Water is then added to fumarate to form malate. This reaction is catalyzed by the enzyme fumarase.
Hydrogen atoms reduce NAD plus to NADH, and malate is then converted to oxaloacetate. This reaction is catalyzed by the enzyme malate dehydrogenase. The cycle is then ready to begin again. Overall, Krebs cycle creates 6 NADH molecules, 2 ATP molecules, and 2 FADH2 molecules for each molecule of glucose.
The electron transport chain is found in the inner mitochondrial membrane in the mitochondria. This chain creates ATP through oxidative phosphorylation. There are 7 main components of the ETC.
The ETC proteins are set up in order of increasing electronegativity. Complex 1 has the lowest electronegativity and complex 4 has the highest. Electrons are then passed from one complex to the next along the membrane. NADH binds to the first complex, which is NADH dehydrogenase, and FADH2 binds to the second complex, which is succinate dehydrogenase. The hydrogen ions are then pumped into the intermembrane space. UQ carries electrons from complex 1 to complex 3. Site C carries electrons from complex 3 to complex 4. Electrons from FADH2 go through fewer electron carriers and make less ATP than NADH. Each NADH molecule creates 3 ATPs, while FADH2 molecules only make 2 ATPs. As the 2 electrons leave complex 4, they combine with oxygen and hydrogen to make water. Overall, ETC creates an electrochemical gradient that can power ATP synthesis.
Hydrogen ions, which are in the intermembrane space, are forced through ATP synthase, which is in the intermembrane. Hydrogen ions then travel into the matrix. The flow of protons creates energy to make a bond between ADP and inorganic phosphate, and ATP is made through oxidative phosphorylation.
Cell respiration is now complete, and 38 ATPs have been made.